My name is Peter ten Dijke, and I'm one of the senior authors of our recent EMBO journal paper in which we examined the interplay between glycosphingolipids and TGF-β signaling in cells undergoing epithelial to mesenchymal transition. It's the result of a collaboration between the ten Dijke lab that is focused on TGF-β signal transduction and the Wuhrer lab that specializes on glycoanalytical techniques using mass spectrometry. Epithelial to mesenchymal transition is a morphological process in which epithelial cells lose their cell-cell contacts and acquire a mesenchymal fibroblast phenotype. The mesenchymal cancer cells show an enhanced ability to migrate, invade, and to form metastases. The multifunctional cytokine TGF-β is a potent inducer of EMT. TGF-β signals via specific cell surface TGF-β type 1 and type 2 receptors. Upon ligand-induced heteromeric complex formation, the TGF-β type 1 receptor phosphorylates and activates intracellular SMAD effector proteins. In this paper, we examined whether the TGF-β-induced EMT is accompanied by changes in glycosylation patterns using a sensitive glycomics platform. Using two well-established cell models for TGF-β-induced EMT, we found that in response to TGF-β, the levels of glycosphingolipids, and in particular the gangliosides, are strongly decreased. The synthesis of glycosphingolipids starts with the addition of glucose to ceramide by the enzyme UDP-glucose ceramide glycosyltransferase, in short UGCG, and continues with the formation of more complex glycosphingolipid structures. Production of gangliosides is controlled by the enzyme SD3-Gal5. We found that TGF-β decreases SD3-Gal5 expression, providing an explanation why we see that in response to TGF-β, the SD3-Gal5 catalyzed gangliosides are decreased. These findings made us look at the possible functional role of these ganglioside changes in TGF-β signaling and TGF-β-induced EMT. When we inactivated the enzyme UDCG and thereby blocked the synthesis of all glycosphingolipids, we found that TGF-β signaling and TGF-β-induced EMT and metastasis were enhanced. In contrast, ectopic expression of SD3-Gal5 inhibited lung cancer cell invasion and metastasis. Next, we investigated the mechanism by which gangliosides inhibit TGF-β signaling and EMT. We found that gangliosides stimulate the redistribution of the TGF-β type 1 receptor from clathrin-coated pits to lipid rafts. Consistent with previous observations, we found that increased localization of the TGF-β type 1 receptor in lipid rafts enhances TGF-β type 1 receptor turnover by stimulating its degradation. Interestingly, when we examined the SD3-Gal5 expression in lung cancer biopsies, we found that its expression in cancer cells was much lower than the adjacent normal tissues. This, taken together with our in vitro and in vivo findings, suggests a possible role for SD3-Gal5 as a tumor suppressor. It will be interesting to explore in future studies the potential of SD3-Gal5 as a biomarker and a molecular target for lung cancer therapy.